Hello to our friends joining us via recording. We are reviewing for the exam for a second time. Remember that the exam is due by Tuesday night at 11:59 p.m. So please make sure to get that done by the 23rd. We have several topics that we want to make sure we discuss today. There is also that previous recording from our session about a week and a half ago talking about this material as well. So make sure to check that out if we miss one of the topics you're interested in. One of the first things that we wanted to make sure that we covered during our class today is hydrogen bonds. So I'm going to start us here. For my friends who are in class with me today, remind me, is this lesson number 3, I think? What page do you have this picture? If anyone can help me out fast. If not, it's not a big deal. We're talking about hydrogen bonds. So hydrogen bonds, as we're searching our notes for them, when you think about hydrogen bonds, one of the first things I want you to make sure that you're thinking is that hydrogen bonds are very weak. So they're very weak. That's the first thing for us to know about them. The second thing for us to know about hydrogen bonds is that they last a short time. Hydrogen bonds last a short time. Okay, perfect. Kaylin says we're on page 4 of lesson number 3. So you find this picture, page 4, lesson number 3. What we're looking at in this picture, this is one water molecule, so H2O, 2 hydrogens and 1 oxygen. This water molecule is attracted to, or it's interacting with another water molecule. When you look at water, water forms hydrogen bonds between those molecules. This is why you can do the experiment like we see a picture of right here. You can take a coin and slowly drip water droplets on top of it. Notice how that water is extending out over the edge of the coin. So this is an idea that we see with water, something called cohesion. Cohesion. The fact that water sticks together, or it's so sticky, if you will, that it will extend past the bottom of the coin. It'll kind of hang out there in space. It's because the water molecules share hydrogen bonds with each other. Now, hydrogen bonds, I think it might be more accurate for you to mentally put them in air quotes. We'll do little air quotes with them here. Because like I mentioned, hydrogen bonds are really weak. Hydrogen bonds are a lot like ionic bonds. Let's see if we can remember with ionic bonds, what is it that holds things together when you have an ionic bond? Why are ions in a bond together when we make an ionic bond? Ionic bond. Yeah, so it is about electrons. Morgan's right. And any of our types of bonds, it's about electrons, right? Yeah, so when we have an ionic bond, we've transferred electrons, right? Or remember, I like to call them stealing bonds. So an ionic bond is a stealing bond, meaning that somebody stole an electron and somebody got an electron. If we're being kinder, we say it's transferring, right? So somebody got an electron, somebody lost an electron. What keeps those ions together is this idea that opposites attract. Opposites attract. If you ever see a test review question or a question on the exam that asks you about the way that ionic bonds work, this is the way they work. It's all about opposites attracting. So when we say opposites attract, we've got 2 things that are being attracted to each other. One of them is called a cation. One of them is called an anion. Which of these things, the cation or the anion, which of these has a positive charge? Is it the cation or the anion that has the positive? Exactly, yes. Several of us are saying correctly, it's the cation that has the positive charge. So it's the anion that has the negative charge. What holds together a cation and an anion, which are the two things in an ionic bond, is the fact that opposites attract. Somebody's always positive, somebody's always negative. I want to be right next to you because opposites attract. Hydrogen bonds are kind of like the little brother of ionic bonds in the sense that what's making them work too is opposites attracting. However, hydrogen bonds, we've got two things that have partial charges. My ions down here, they've completely transferred an electron. Somebody's totally positive, somebody's totally negative. In a hydrogen bond, we've got a couple of partial charges that are attracting to each other. Can anyone remember, what do you call that part of a molecule that has a partial negative or a partial positive? Yeah, a couple of us are remembering. Here's one of our really important words from unit number 1. The word is a dipole. Dipole. So when we talk about a dipole, it's a partial positive or negative. Partial positive or negative. I can see when I'm looking at my water molecule right here by my hydrogen, we've got a partial positive right here by the hydrogen. And up here is another water molecule. Here's my oxygen and my 2 hydrogens. This part of my molecule is the oxygen with its partial negative charge. So hydrogen bonds form when this partial positive is attracted to this partial negative. It's still this idea that opposites attract. But because we're just doing dipoles attracting to each other, and we're not doing ions attracting to each other, this is a really weak bond. It lasts just long enough to help water molecules not fall off the edge of the coin, but it's not going to last forever. So hydrogen bonds are the weaker younger brother of ionic bonds. They form because water has dipoles. Hey, let's make a connection. I have an atom or a molecule here with hydrogen and oxygen in it. And hydrogen and oxygen, when they form bonds with each other, they make dipoles. If I make dipoles, I am not sharing my electrons equally. That's how I got a dipole. I'm not sharing equally. So help me out. I should be able to predict the name of the kind of chemical bonds that I have. Which kind of chemical bonds help me form dipoles when I'm not sharing electrons equally? We're not sharing electrons equally. Yeah, so polar is my tip off for unequal. Yeah, and then my word for sharing, remember, is covalent. Covalent. So in molecules that have polar covalent bonds inside of them, covalent always means sharing, polar means unequal. If I am not sharing my electrons equally, somebody is partially positive, somebody is partially negative. I have dipoles. I have areas where we're not sharing equally. And if we have dipoles, it's highly possible that we're going to form hydrogen bonds. Hydrogen bonds are what happens when a hydrogen, which always has a positive dipole, is attracted to a negative dipole. So we're going to see next unit when we start talking about your DNA. Sometimes hydrogen is attracted to nitrogen instead of oxygen. So what holds the two halves of your DNA double helix together is actually hydrogen bonds. They connect the two sides together. Sometimes this positive dipole on hydrogen likes to interact with a nitrogen or a sulfur. But when we say hydrogen bond, it always means that a positive dipole in hydrogen is attracted to a negative dipole. So remember, dipoles, partial positives or partial negatives. The way I form those is with polar covalent bonds. Polar covalent bonds. Hey, we also talked about kind of the opposite of polar covalent bonds. We also talked about non-polar covalent bonds. How would we describe what happens in a non-polar covalent bond? What's going on with my electrons in a non-polar covalent bond? Yeah, so we still got that covalent word. And remember that covalent always means sharing. We're sharing our electrons. And that nonpolar word, nonpolar means now we're sharing them equally. So a nonpolar covalent bond is an equal sharing bond. Equal sharing. This is the way that it's supposed to be, right? We talked about how every parent wants their kids to do nonpolar covalent bonding to share things equally. If we're lucky, it's polar covalent bond where we unequally share things, but at least we share. But if we're more honest, there's probably a lot of ionic bonding going on, right? One child steals it and now the other child wants it. But non-polar covalent bonds, that's best case scenario. What can you tell me, by the way, about non-polar covalent bonds? Are they strong or weak? Non-polar covalent bonds, are they strong or weak? Yeah, these are, like Kaylin said, these are the strongest. Absolutely. This is the strongest type of bond that we make between things. If we are equally sharing our electrons, that's the strongest way that we can bind things together. Remember though, anytime we're sharing, so anytime we have this covalent word on there, it is still a relatively strong bond. So these ones still are pretty strong too, polar covalent bonds. Even though we're not sharing equally, we are still sharing. So that's a pretty strong bond. Ionic bonds are only based on this idea that opposites attract. So ionic bonds are going to be quite a bit weaker compared to the covalent bonds. Who can remember what happens to an ionic bond or a molecule that's made with an ionic bond when I put it in water? What do ionic bonds do when there's water around? Yeah, exactly. Like Regina and Kaylin said, if you put something that has an ionic bond in it, if you put that in water, so if you take your salt shaker and shake some salt into a cup of water, that salt's going to completely fall apart. Opposites attract. Those different charges are what holds them together. If we bring in other charges, I'm just as attracted to those as I was to you. So ionic bonds can't survive water because it's all just about those opposite chargings attracting. With these hydrogen bonds, we see them inside water, but they keep forming and breaking apart and forming and breaking apart because they last such a short time and because they're such a weak bond, because it's just dipoles being attracted to each other, these ones are going to be very weak as well. I will not ask you on the exam, by the way, to put things in order of how strong they are. But what you do need to know is the strongest is the non-polar covalent bonds. And of the ones that we're talking about, the weakest is going to be those hydrogen bonds. If I were to rank them, I'd say number 2 with our polar covalent and our ionics being number 3 down there. When we talk about polar covalent bonds, so being in a molecule that has dipoles, remember that we looked at this picture, actually a couple of pictures. This molecule over here on the left, notice how we've got this weird squiggly letter. So this is that symbol for dipole, what we were just talking about. This part of my molecule has a positive dipole. This part of my molecule has a negative dipole over here. So when we have a molecule that has dipoles, like this one we're looking at, remember from the guided lesson, what happens is a water molecule comes over. And remember that water molecules have dipoles as well. So my water molecule has its own negative over here and its own little positives up here. So my water molecule comes over and says, hey there, negative talking to the positive. And this water molecule down here has its own positives going on. So we got a little positive down here. This one down here is saying, hey there too. What happens is the dipoles in this molecule will start to form hydrogen bonds with water. So what your picture doesn't include that we could add, now that we talked about it, is you could put some little dashes right here. I've got a hydrogen bond that forms between the hydrogen in water and the negative dipole in this atom. So this one's got a negative dipole. It has a partial negative charge. Water's got a partial positive right here. These will actually form a hydrogen bond with each other. Now, this molecule is not going to split apart. We're not going to fall apart like happens with salt. But we are going to interact with those water molecules. So we form a hydrogen bond here. Remember the term that we talked about? We said that we play nice with water. Does anyone remember that long chemistry word for I play nice with water? I play nice with water. Oh, it's a big, long word. It's mean for me to make you type it. Yeah, the chemistry word for I play nice with water is hydrophilic. Hydrophilic. Here's let's make a connection right here. If you are hydrophilic, you have dipoles, partial positives and partial negatives. You want to know why you play nice with water? Because you can make hydrogen bonds with water. Let me say that again. If you play nice with water, you've got a positive dipole and a negative dipole. You are polar. Things that play nice with water are polar. The reason you play nice with water is because you can form a hydrogen bond with molecules of water. So one of the things that we said we wanted to review was what did and didn't play nice with water. Hydrophilic things are the things that do play nice with water. One of the words we just added to that list is polar, right? Polar meaning I've got dipoles. So if you've got dipoles, you make hydrogen bonds with water, you play nice with water. Hey, we talked about on the previous slide, we talked about our ions. If you have a fully positive or a fully negative charge, you play nice with water. The one other word I'll add to our list that's kind of similar to it is water soluble, but that's kind of like a cheating one, right? If you're water soluble, you dissolve in water. So obviously you play nice with it, right? But these were our words that meant the same thing. Hydrophilic and polar, water soluble, all of those go together in a group. What we see happening in this second picture here on the slide is when we have a molecule that does not have dipoles, so when we have a molecule that does share their electrons equally, when water comes over with its partial positives and partial negatives, these molecules don't want anything to do with that. These molecules are sharing their electrons equally. So when you come over with the partial positive or a partial negative, these molecules say, hey, I don't need that kind of negativity in my life, or I don't need that kind of positivity in my life. Our charges are balanced. We're equal over here. So these are the things that when we say they don't play nice with water, what's happening is water really wants to try to form a hydrogen bond with them, but water can't form a hydrogen bond with them because these ones have no dipoles. They don't have a partial positive or a partial negative for water to interact with. Let's add to our list over here. So we just now talked about things that are hydrophobic, things that don't play nice with water. What were some of my other words that I would use to describe things that are hydrophobic? What are my synonyms for hydrophobic? Yep, Elipina listed one of them for us. Non-polar, I'll put non in caps, non-polar. Absolutely, I'm scared of water, right? So afraid of water, that's what that word literally means, right? And the other one that we'll add to the list as well is that we are lipid soluble. Lipid soluble. So if you are afraid of water, you are non-polar. You've got no positives, no negatives. You do great with lipids because lipids have non-polar covalent bonds. You don't do great with water because water's got those polar covalent bonds. We had a question in the chat that someone answered for us. I want to make sure to cover it too, though. Let me see if I've got the picture I'm looking for. Otherwise, I get to go all artsy on you. Okay, perfect. The question that we had in the chat is basically which of those sets of words are things that can get across the plasma membrane. So here I am looking at a very simplified plasma membrane right here. Can you remind me in the chat, we've got the heads up here that we see in our picture, that's these blue circles here, and we've got the tails that we see here in the middle of the membrane. Is it those heads or is it the tails that we would say are hydrophilic? Heads or tails? Who's hydrophilic? Who loves water? Yeah, exactly. The heads love water. So here we go. I've got my water-loving parts. So we are hydrophilic. We love water. And my tails then are hydrophobic, right? So, oops, let's fill that in. Hydrophobic tails in the middle. Hydrophobic. Remember from when we filled in information in this picture, remember that there's some water inside the cell. Oops. That is what happens when you press shift with the 0. H2O, try again. There's water in the middle of the cell. There's water on the outside of the cell. Where there is not water is here in the middle, in this part that's hydrophobic. Because this part here in the middle exists, or the reason that our cell makes a membrane with 2 layers is to make sure there's no water in here. So if you want to get across, if you want to sneak from outside to inside, you're going to have to match this hydrophobic middle part here. So you're going to have to use some of our words from before, you're going to have to be lipid soluble, for example, because it's the lipid tails that are in the middle of the membrane. Or you're going to have to be non-polar. You're going to have to share your electrons equally to be able to sneak through. And remember, we had this chat, what feels like an eternity ago. I'm promising you that on the exam, there is going to be a question where I ask you what can and cannot get across the membrane. And I'm going to give you crazy chemicals that you've never heard of before, or I'm going to give you crazy names of proteins or something that looks super scary. But right behind those super scary words, you're going to see something like, this is hydrophobic, or you're going to see something like, this is polar, or this is water soluble. Look for those words behind their super scary names. I promise that question's not as hard or as scary as you feel like it is, but I want to make sure that you can use your information. So when we talk about what can get across the membrane, remember that you have to match the middle of the membrane. We have to be hydrophobic. We have to be lipid soluble. We have to be non-polar. Or there was one other word. Does anyone remember? It's not really a chemistry word as much as a size word. Is it really large things or really small things that can get across? Who can get across, large or small? Yeah, it's those small things. Small things can get across. So here we'll build out our list for ourselves here. If you're hydrophilic, that also means you're water soluble. Water soluble. That also means that you're polar, and as opposed to small, you're probably big. The other word for things that can't get across the membrane are big things too. How do we feel about what can and cannot cross the membrane by itself? Thumbs up, thumbs down. How do we feel? Uh-oh, I think I've lost you. There come a few thumbs up. So Caitlin asked a good question. She said, if you're hydrophobic, are you also small? Not necessarily. This is kind of its own thing. So here, let's mark it more accurately. Number one is all these groups of words. These, if you say one, the others are true as well. Here's the other characteristic of something that could get across the membrane. If you're small, you might still be polar, but if you're really small, you can sneak across. If you are non-polar, you can actually be somewhat large and still get through. But if you are, if you're really big, so even some hydrophobic things that are really big, they're not going to get across either. So you can't be insanely huge, but you definitely are going to have to be one of these things. So all of these words mean the same thing. All of these words mean the same thing. And then our size characteristics are kind of their own thing there. So good clarifying question. Okay, so let's talk about, for all my molecules that are water soluble or that are polar, that need to get into my cell, I need to still be able to make that happen. And those lipid tails aren't going to let it happen on its own. So this is where proteins come into play. When I need to get something across the membrane, here's your hint for you right here. That's lipid insoluble. Lipid insoluble. If I am insoluble, that means I don't play nice with something. If you're lipid insoluble, you don't play nice with the lipids in the middle. which means, yeah, like Elapina said, we would be hydrophilic. If we don't play nice with lipids, we play nice with water. Remember, I mentioned this forever ago. If we have to go through a protein to get across the membrane, we must be lipid insoluble. Or to say that a different way, if you're lipid soluble or if you're hydrophobic, you're not going to get into the cell using a transport protein. We only use transport proteins for things that can't sneak inside. If you match the middle of the membrane, you can just sneak in and out. You don't have to use a protein. It's only if you don't match that we have to use a protein. Now, we see the two types of proteins that are used to move things across the membrane in our picture here. I've got carrier proteins and I've got channel proteins. Can you help me out in the chat? What makes a carrier protein different from a channel protein? How is a carrier protein different? Yeah, so Kaylin gave us one of the ideas. Carrier proteins, they change their shape, absolutely. So we can see that in the picture. We can see it going from open on one side to open on the other side. Carrier proteins, they change their shape. So carrier proteins, as part of their shape change, one of the other things we talked about is they attach to what they're transporting. So notice in our picture, I'm open to the outside, and what I'm going to transport comes in. I attach to it in this special place for it. Then I change my shape, I open to the inside, and I release it. Carrier proteins attach to what they're moving. They open and close on different sides of the membrane, one side at a time, like Elapina mentioned, absolutely. When we talk about channel proteins always have a channel. They always have an open space in the middle. So when a channel is moving something, it never attaches to that something. The something just passes straight through it. But Zaira mentioned a good point for us. That's true with some of my channel proteins. Some of those channel proteins have a gate on them. So some of them I've got to have the right kind of thing. So whether it's a chemical thing, whether it's a pressure thing, whether it's a charge thing, I have to have some kind of stimulus to get it to open or close. So when we talk about channel proteins, all of these kinds, here's something to keep in mind every time we're thinking about a channel protein. Big picture, really important idea. Channels don't attach. I really should have just typed that, shouldn't I? Channels don't attach. I don't care if you look really easy, we're always open, or if you open and close. Any of my kinds of gated channels, or what we call the leakage channel, they're never going to attach to what they're transporting. So when we look at this channel over here on the left, the one on the left is called a leakage channel. This is what we saw on the previous screen, how it's always open, and I've always got this space for stuff to move through. We never attach to what we're moving, and in the case of a leakage channel, we're always moving stuff. Hey, remind me in the chat, with a leakage channel, do we transport things from low concentration to high concentration, or from high to low? Low to high or high to low? What do I do with a leakage channel? Yeah, so we are doing facilitated transport. We're doing facilitated diffusion. I probably should have said that word before. All of these are an example of facilitated diffusion. Let's remind ourselves when we talk about diffusion, which way do we go in diffusion? From high to low or low to high? Which way do I move stuff in diffusion? Yeah, we actually got a little bit mixed up. We can blame the snow. It's totally cool. Uh, Diffusion is an example of passive transport. So it means that it's passive transport, so there's no energy. No energy, which means we're always going to go from high concentration to low concentration. We're always going to move stuff from where there's a lot to where there's a little. So notice with my leakage channel here, I've got a lot of this lipid insoluble thing outside the cell, and I'm moving it into the cell. Leakage channels are always open. They always move stuff from high concentration to low concentration. My other kinds of channels that I see over here, all three of these kinds of channels that I see over here are called gated channels. Gated channels. Remind me in the chat, gated channels are or are not always open. Gated channels are or are not always open. Yeah, exactly. Several of us have mentioned this. Gated channels are not always open. It's that puppy analogy that I gave you, right? We've got puppies in the backyard. The gated channels are the gates that are holding them in. So these are not always open. They're not always open. It takes different things to open up these channels, but when any of these kinds of channels open, we are still always doing facilitated diffusion. We're still always going from high concentration to low concentration. So up here, it's the binding of a chemical. We use a chemical key to open up these channels. We would call this a chemically gated channel protein. Chemically gated, because we use a chemical key. When I look over here on the right side of my image, notice that this channel protein is closed. Here's my gate. The gate is closed when there's a negative charge inside and a positive charge outside. When it becomes positive on the inside, my inner membrane, that gate opens up. This is a channel that opens because of a change in charge. We would call this kind of channel a voltage gated channel. A voltage gated channel. And my last kind of channel that I see down here opens with pressure, or it opens when I push on it. I would call that a mechanically gated channel, because I use what's called mechanical energy to press on this gate to push it open. But here's an important idea. Please make sure we know this. I know we've said it before, but we're going to say it again. Anytime we have a channel protein, whether it's a leakage channel or a gated channel, any kind of channel protein always does facilitated diffusion. We always go from high concentration to low concentration. If I'm always going from high to low, I never use energy. So all of my channel proteins, all of my gated channel proteins, they don't need energy to work. They just need the right key to open the gate for them. We had a question about some of those transport words. I believe you have this picture in your notes somewhere. Did I give you this picture in your notes? Or is it just in my notes? I don't know if we've seen this one before. Don't think so. Tian says this looks new, possibly. Well, hey, we get to look at a new picture. Let's see if we can make sense of it, even if we don't have this in our notes. My picture here shows us the process of diffusion, facilitated diffusion, and active transport. When we talk about the process of diffusion, diffusion is when things go straight through the membrane of the cell. Diffusion is an example of passive transport, which means that using the process of diffusion is always going to take me from high concentration to low concentration. This part of my image shows me the process of diffusion, from high concentration to low concentration. When I look at the second part of my image, I'm looking at facilitated diffusion. In facilitated diffusion, we are using a protein facilitator to do the exact same thing that we did in diffusion. So we're still going from high to low, but we're doing it through a protein channel. So facilitated diffusion uses a channel protein to move things from high concentration to low concentration. When we talk about active transport, we have to use energy to do active transport. And the reason active transport takes energy is because we're going from somewhere where there's not a lot of something, and we're taking it to where there's already a lot of it. Active transport, we're going uphill, or we're moving things from low concentration to high concentration. That's why it's active transport. That's why it takes energy to do this kind of movement. When we talk about the kinds of passive transport, there's one other kind of passive transport that didn't make my diagram. Let's go ahead and add it at the top up here. The other kind of passive transport that we talked about together in our class is osmosis. Osmosis. Can you help me out in the chat? What happens in the process of osmosis? What's going on in osmosis? Exactly. Yep. Osmosis is all about the solvent. In the body, it's the solvent that we care about is water. But to use our chemistry word here, we'll all put solvent here in quotes. That's the liquid we dissolve stuff in, the solvent. So in the process of osmosis, we are moving the solvent, which most of the time in the body is water. When we talk about the process of osmosis happening, we're still always going to move from where there is a lot. So I'll draw some water molecules here. From where there's a lot of water to where there's a little bit of water. Osmosis is also passive transport. Always going from high concentration to low concentration. The caveat being high concentration of water to where there's a low concentration of water. By the way, we're looking at the inside of the cell right here and the outside of the cell right here. What's the name of the soupy stuff inside the cell? What's the soupy stuff inside the cell? Yeah, exactly. Remember the name of that fluid, cytosol. That's the fluid inside the cell. If I have less water inside the cell than I have outside the cell, water is going to want to come in. That, by the way, is what happens in water intoxication. When there's a lot of water outside, not as much inside, it's going to do osmosis and go into the cell. Yeah, so Kaylin got where I was going with that too. So if I've got a lot of water outside compared to inside, I would say that the solution here on the outside, my word for it is hypotonic. Hypotonic. That means I've got a lot of water and not a lot of dissolved stuff. So a hypotonic solution has a lot of water and not a lot of dissolved stuff. Hey, if the stuff on the outside has more water, that means the stuff on the inside has more salt. What's the word that's the opposite of hypotonic? What's my opposite word from hypotonic? Yeah, exactly. My opposite word is hypertonic. Hypertonic. Remember that we always use these words to kind of compare things to each other. So my cytosol can be hypertonic, has less water, while my solution on the outside is hypotonic. If that's the case, my water is going to come inside. It's going to try to dilute this. What if there's the same amount of water on the inside and the outside? What's the word I would use then if the amount of water is the same? Exactly. Isotonic. That's when it's the same inside and outside. This is what our body likes. We would much prefer that our plasma, that's the fluid outside of red blood cells, we want it to be isotonic to our cytosol, that is inside red blood cells. Because if that's the case, the amount of water that comes in is the same as the amount of water that comes out, and our red blood cells stay the same size. If that plasma gets hypotonic, then we got that water intoxication stuff going on, and that's no good for us. I've talked a bunch. I need to pause for a moment. Can you tell me in the chat what questions we have about anything we've talked about so far? Okay, so Kaylin has a good question that I'm going to pull up that picture from our notes. Let me see if I have it. Yes, right here. Yeah, so Kaylin's asking specifically about red blood cells. So I'm going to ask you all to help me out with this. I'm looking at right here, the amount of water going in and out is equal. So if the amount of water that goes in and that goes out is equal, what's my word for this first blank here? Oh, only one of us know. There we go. More answers coming in. You were totally right, Anisha, but I wanted to see if anyone else knew too. If we've got the same amount of water going in and out, that's isotonic. When I see water only going in to my cells, so much in fact that my cell might explode because too much water is going in, what would I call this solution out here if it makes water go into the cell? Yeah, exactly. Hypotonic. Hypotonic. When water goes into the cell, that's because there's a ton of water on the outside of the cell. Remember, we had that phrase that water follows salt. Wherever there's more salt, So wherever it's saltier, water is going to move that direction. There's more salt inside than there is outside. That water is going to rush inside. So if I've got more water, I'm hypotonic. Over here, the opposite's happening. See how that water is leaving the red blood cells? When the water leaves the red blood cells, they shrivel up and they get smaller because there's more salt, or there's a higher concentration of salt on the outside. What word do I use here if I've got a higher concentration of salt on the outside? Yeah, higher concentration is hypertonic. Hypertonic. So higher concentration, hyper, lower concentration, hypo. If I have a lower concentration of salt, that means I've got tons of water. And all that water wants to rush inside. That water wants to go into our cell. Does that help with the question that we had? So the short answer, isotonic, water enters and leaves at the same rate. Hypotonic, water goes in. Hypertonic, water goes out. Yeah, Kaylin says she thinks her brain froze this last week. I feel ya. I'm thinking some of the same things. Absolutely. We need to get a little more of that coffee, right? Get some of that, a little more of that coffee. See if that wakes it up a little bit. Okay, I'm looking at my list here. I want to make sure that I take time to talk about the two kinds of active transport, because we have not talked about that yet. And then we will see what other topics we want to cover. So I'm bouncing all over the place. I apologize. I think we're back to lesson number 4 now with our two types of transport. Can anyone tell me which page are we in lesson 4 here to talk about those transport types? Active transport types, I should clarify. Caitlin says, I remember this. Hey, that's good. At least you remember something, right? Well, so as we're looking frantically through our notes for this here, remember that when we talk about active transport, whether it's primary active transport or secondary active transport, as we'll see in our next picture, both of these are examples of active transport. When we use that word active transport, what does active mean? What are some of the ideas that come with active transport? Yeah, Regina hit a really important one. It uses energy, absolutely. Yeah, and it uses energy because we're going up the gradient, up the gradient. So remember, we literally talked about thinking about how we move things as a hill. So if I've got a hill, I can either go from high concentration to low concentration, that's going down the hill, or I can go from low concentration to high concentration. That's going up the hill. If we are doing active transport, we're going up the hill. So anytime I ask you about the way stuff moves in active transport, we're always going from low concentration to high concentration. We're pushing that rock up the hill. Yeah, so we're on page 4, perfect, of lesson number 4. Excellent. So primary active transport, they're in lesson number 4. Primary active transport, we need energy for, just like secondary active transport. But in primary active transport, what kind of energy do we use to make this happen in primary active transport? Yeah, in primary active transport, my form of energy is ATP. ATP. That's my energy money that the cell builds. To do primary active transport, I use the energy money that's stored inside my cell. So the sodium-potassium pump is an example of a carrier protein that uses ATP to move stuff. When I look at my picture here, something we're just going to have to memorize or know from our picture, in the sodium-potassium pump, which way does sodium move? Is sodium going in or going out in the sodium-potassium pump? Yeah, so I can see my little arrow. Here's my sodium. Sodium is going out of my cell in the sodium-potassium pump. I'm pumping it out. And at the same time, potassium is going in to the cell. So sodium is pumped out, potassium is pumped in. We get to do my favorite, right? You didn't actually think we'd get through class without one of these, right? Salty banana time. Let's start with sodium, because sodium is the first part of this pump. Do I have a lot of sodium inside or outside? Where's the sodium in a cell? Inside or outside? Yeah, sodium, at least one of us remembers. Sodium on the outside. It's a salty banana. So we've also got chloride. That's the other part of salt, right? Chloride, both of those are on the outside of my cell. And my banana has potassium inside of it, just like a cell, lots of potassium. So here's what's happening in the sodium-potassium pump. I'm taking potassium that's outside the cell, and I'm pushing it inside the cell. And I'm taking any sodium that was inside the cell, and I'm pushing it outside. I am making things go the direction that they already are. So my red arrows here are showing us what happens in the sodium-potassium pump. Any sodium that's in here, I'm pushing it out. Any potassium that's out here, I'm pushing that in. If I didn't use energy, remember that stuff always goes from high concentration to low concentration. So if I didn't use any energy to move sodium, if I let it do its own normal thing, what sodium would do is it would sneak inside my cell. Sodium wants to come inside. And what potassium wants to do is it wants to leave. But when I do this sodium-potassium pump thing, sodium moves in, or excuse me, moves out. Ignore Dr. Ahles, not enough coffee, right? Sodium moves out. Potassium moves in. Both of these things are going up the hill. They're both going the wrong direction. Because they're both going the wrong direction, not only do each of those movements need energy, but we need so much energy that I'm going to have to use ATP. Yeah, both of these things are going against their gradient, or both of these movements are going uphill. I need energy to move both of these things the way that they're going. So the energy I have to use is ATP. In primary active transport, I've got to use ATP to move things because both of my things move uphill. They're both going from low to high. When I talk about secondary active transport, it's still active transport. So I still use energy because it's active transport. But when we do secondary active transport, I'm not using ATP anymore. When I do secondary active transport, I am using the energy of another molecule. So I think the way that your guided lesson describes it is another molecule's concentration gradient. Another molecule's concentration gradient, or something like that. The easy way to think about secondary active transport is either piggybacking, or you could think of it like hitchhiking. In other words, somebody else is moving already. They're walking down the hallway already, or somebody else is driving down the road already. I'm just going to use the fact that they're moving, and I'm going to move with them. So in secondary active transport, please never tell me that ATP is used. We never use ATP to do secondary active transport. We're always using somebody else's movement, somebody else going downhill. I can steal that and use it to make me go up the hill. So when we look at our example of secondary active transport, we're talking about the sodium glucose cotransporter. The sodium glucose cotransporter. Now, with the sodium glucose cotransporter, notice that sodium is moving into the cell and glucose is moving into the cell as well. Both of these molecules are moving into the cell. But when we talk about sodium and glucose, one of these was already really concentrated inside the cell. What does the cell already have a lot of on the inside? Yeah, exactly. The cell already has a lot of glucose. Exactly. Glucose is its favorite food, 100%. And cells really like to hoard glucose. They're going to grab any that they can find. So I've got sodium coming into the cell, and I've got glucose coming into the cell. Glucose is going from low concentration outside to high concentration inside. Because glucose is going from low to high, it needs energy. The energy that it's going to use is sodium. Remember, with our salty banana cell, draw it really fast one more time. We've got a whole lot of sodium on the outside. When I leave sodium alone, it wants to go in. In fact, it wants to go in really, really badly because there's so much sodium outside and there's so little inside in comparison. Sodium really wants to get inside. So we've got this protein in the membrane, this carrier protein in the membrane. Sodium sneaks inside its little pocket, and there just so happens to be a hole that's shaped just like glucose. And the cell ignores the fact that it's already got a lot of glucose inside because it so badly wants that sodium to come back in. So glucose and sodium both attach to this carrier protein. It opens up inside and lets both of them inside. It doesn't take energy for sodium to go in because sodium wants to. There's a ton of it outside. But it is going to take energy to get that glucose to come inside because there's already a lot of glucose inside. Remember this big picture idea that we talked about before. If a cell uses energy to do something, it must be important. If a cell uses energy to do something, it must be important. In the case of the sodium glucose cotransporter, this is how I get glucose into the cell. This is how my cell gets the food that it wants, how it feeds itself, is by using the energy of moving sodium back inside the cell. When we talk about the sodium-potassium pump, where sodium goes outside and potassium comes in, what did we say was the reason that a cell uses energy to do this? Why is a cell willing to spend ATP to pump sodium out and bring potassium in? Yeah, Elapina's right. She used a technical word for us that's totally correct. This is what we use to polarizes, can't spell, the plasma membrane. This polarizes the plasma membrane. That's my fancy anatomy word. If a cell is polarized, what does that even mean? What's happening if a cell is polarized? Yeah, it's alive. So Kaylin, I would say something slightly different than balanced. It is normally polarized. So in that regard, yeah, so I like where Zyra went for us here. When we talk about polarized, we actually have different charges inside and outside. This is what we want. We want to have different charges inside and outside. In that way, we are balanced, if you will. So we need to be different inside versus outside to keep us alive, to allow us, when we think about neurons, to talk to each other, to allow us to do some of these types of transport processes. We have to be polarized. We have to be different inside compared to outside. It's so important that we're polarized that a cell will spend ATP to do it. And it's so important that we have glucose, that a cell is going to wreck this work that it does here to push out the sodium to help it get glucose back inside. Both of these, the sodium glucose cotransporter and the sodium potassium pump, both of these are examples of active transport, of the cell getting stuff that it needs or setting itself up to function correctly by using energy. How do we feel about this table? Yeah, Elapina made a good point, and I'll put that note here. You have this table in your lesson, by the way. We've kind of talked through all this stuff. I'm happy to work through it if we'd like, but I kind of want to gauge and see where we're at. Both use carrier proteins when we talk about these transport processes. Okay, so we can work through it really fast here. Sodium glucose cotransporter and the sodium potassium pump, both of these are examples of active transport, but they're both specific kinds of active transport. When I talk about the sodium glucose cotransporter, was that primary or secondary active transport? Primary or secondary? with the sodium glucose cotransporter? Yeah, a couple of us are chiming in. This is secondary active transport. Secondary active transport. True or false? In secondary active transport, I use ATP. Secondary active transport. True or false? It uses ATP. Yeah, that is false. Correct. Secondary active transport, no ATP used. We use the movement of somebody else. So when we think about the sodium glucose cotransporter, which is secondary active transport, let's talk first about why the cell wants to do this. The cell wants to do this for glucose. Which way is glucose moving? In or out? Which way does glucose move in the sodium glucose cotransporter? Yeah, exactly. Glucose moves into the cell. I'm putting parentheses here because it's food. Glucose moves in because it's food. That's the movement that takes energy. So my energy is coming from sodium. Oops, let's try that again. My energy is coming from sodium. Is sodium moving in or out? Which way is sodium moving? In or out? Yeah, sodium is also moves into the cell. When sodium moves into the cell, it's going down its concentration gradient. So when sodium moves into the cell, it's going from high concentration to low concentration. It's going from outside where there's a lot to inside where there's not very much. This is the energy. So sodium is my energy source. This movement is my energy source. Energy source. Because sodium goes down its concentration gradient, this is the energy. Compare that to the sodium-potassium pump. In the sodium-potassium pump, which way is sodium moving in that one? Which way does sodium move in the sodium-potassium pump? I know I switched sides on you, sorry. Yeah, in the sodium-potassium pump, sodium moves out of the cell. And at the same time, potassium, yep, just like Elapina said, potassium is going to move into the cell. Absolutely, potassium moves in. Both of these ions are moving uphill. Because both of them are moving uphill, I have to use ATP. So ATP is the energy source. ATP is the energy source. So yes, the sodium-potassium pump, we would say that this one is an example of primary active transport. Primary active transport. I can't use the energy of somebody going downhill because both of my somebodies that are moving are going uphill. Both of them are going up their concentration gradient. So I have to use ATP to do the sodium-potassium pump. But like we said, the reason that I'm willing to use ATP to do this, my overall purpose of this, This is what I use to polarize the cell's membrane. This is what I use to make sure that the inside is different from the outside. And remind me, with the sodium glucose cotransporter, what was the purpose of that one again? Why do I want to have that for my last question over here? Why would I do the sodium glucose cotransporter? Why is that one good? Yeah, to get glucose, or in easy words, to feed the cell. To feed the cell. Absolutely. Yep, stock up on that sugar, right? I think you said it before, Kaylin, right? That you like your sugar too. I'm right there. So we got to stock up for the next blizzard, man. Get that sugar. Sodium glucose cotransporter. I'm all on board for that. All right, so I'm looking at my list, and I think all of the topics that I have written down, we have covered. We still have some time left. Are there other particular topics that we want to cover with the time we have left? Good question, Regina. Regina asked about what I like to call the recipes of the fluids. Yes, that is something that you need to know. So let me pull up our, let me see if I have that. Okay, I have a slightly different version of that graph from where we first talked about that. Let me pull it up here. What we don't see on this version, alas, is proteins. But this version can help us out when we're answering most of the questions. Yeah, exactly. So when we're working on the recipes for the fluids inside and outside the cell, you first have this graph. I think it's in lesson 2 that you first have it. So go back and reference this graph, or yeah, like Elapina's mentioning, this is why I keep talking about the salty banana, because it's the easiest way to memorize what I have a lot of in different places. So when we're talking about interstitial fluid, or when we're talking about plasma, my bar graph here, any places that have a big blue bar, that's a lot of stuff in the interstitial fluid. Any places with a red bar that's big, there's a lot of that in plasma. Notice that I have really big blue and red bars here for sodium and chloride. So when we're thinking about a banana that we sprinkled some salt on, we put that sodium and the chloride, that makes salt, on the outside of our banana. So we need to know that there's a lot of sodium and chloride outside the cell. We also need to know that on the inside of the cell, with this beige bar here, we've got a whole bunch of potassium. So when we keep talking about our bananas have lots of potassium inside of them. Intracellular fluid has a lot of potassium. So cytosol has a lot of potassium. The other two things that I don't see on this version of the graph, one of them we just talked about, glucose. Do I have a lot of glucose inside or outside? Do bananas have a lot of sugar? Yeah, I got a lot of glucose inside, right? Because bananas are sweet. So lots of glucose inside my cell. And remember, we kind of talked about how our cells are like the perfect banana, how the perfect banana would also have a lot of proteins in it. So there's a lot of proteins inside your cells. One of the biggest differences when we compare interstitial fluid versus plasma is the level of proteins in these two types of fluids. So plasma has a lot of proteins. Interstitial fluid, I'm just going to put few, very few comparatively. So interstitial fluid and plasma look really similar in that they both have a lot of sodium and they both have a lot of chloride. But what's the biggest difference between them is that interstitial fluid doesn't have very many proteins, whereas plasma has a lot of proteins. So we do need to know those recipes. Salty banana hits most of those parts. And then just knowing the difference in the number of proteins in these two types of fluids. I'm looking back at my chat here. So Kaylin asked the question about the sodium potassium pump going from low to high. Also, that is correct, Kaylin. Both of the molecules in the sodium potassium pump, they're both going from low to high. So since they're both going from low to high, that's why I have to use ATP, because they're both going uphill. We got a question about carrier proteins and channel proteins. So for my friends here in class, help me out. The question is, does a carrier protein or a channel protein transport things that have polar bonds? Or in other words, transport things that are polar? What do we think? Yeah, channel moves faster. That is correct. Channels are faster than carriers. If I am a polar molecule, can I get across the membrane by myself? Can polar molecules get across? Yeah, polar molecules can't get across by themselves. Polar molecules need help. If I need help, I can go through either a carrier protein or a channel protein. I can use both of them to get across the membrane. So carrier proteins and channel proteins. I use both of those kinds of proteins to transport polar things. I could also use carrier proteins and channel proteins to transport ions. What I would not use carrier proteins or channel proteins to transport are going to be non-polar things. If you have no charge, if you are lipid soluble, I wouldn't use any kind of protein to move you across the membrane because you can just go straight through on your own. So when we're talking about whether I'd use a carrier protein or a channel protein to move things, because I know you have some review questions about that. With both carrier proteins and channel proteins, the big idea is I'm moving things that are polar, things that are lipid insoluble, all of those chemistry words of the stuff that can't sneak across the membrane. Channel proteins and carrier proteins can both move those kinds of things across the membrane. That's an example of Dr. Ahles being tricky, trying to make you think there. What other questions do we have? Yes, you're welcome. All right, well, I'm not seeing any other particular questions. Before I end the recording, let me just remind you about a few things about the test, just so we are aware of it. So exam number 1, let's talk a little bit about this just to make sure we're good on it. First thing we should know about exam number 1 is you have 75 minutes to complete it. Minutes to complete it. I think it has like 57 questions on it, so that's more than a minute a question. Take your time and read those questions slowly, then pick your answer and move on. This exam needs to be submitted, so we need to have finished it by 11:59 p.m. Please plan to take it so that you have more than 75 minutes before the deadline. Otherwise, when you finish it, it's not going to give you a score. I'm going to have to go in and manually grade it. So make sure it's done well before midnight tomorrow night. Let's see what else. Before you can take the exam, so before you take the exam, you have to complete the Proctorio practice test. Proctorio practice test. It's literally just one true-false question, but if Proctorio is not working on your computer, you're not going to be able to open it up. So you need to install the Proctorio extension on your computer. There's a link for it with directions in the lecture exams folder. So first, install Proctorio. Then try to take that practice test. If you are asked for a password, so asks for a password, if that is what happens to you, what that means is that your Proctorio extension is not working. So Proctorio isn't working. I don't have a password for the exam. I can't let you in. Proctorio makes that password once it's logged in on your computer. So part of why I'm asking you to do this practice test is to make sure that Proctorio is working. If you go and click on either the practice test or the regular test, and one of them is not letting you in, there's a whole document about how to get Proctorio working. So work through, it's like 11 different ways to make sure Proctorio is working. Work through those ways. At least one of them should work for you so that you can click on that exam and get going. Like I mentioned, you've got 75 minutes to work your way through it. Should be more than enough time. I think students usually take it in 40 minutes or less. So you've got plenty of time for this exam. Remember what we talked about way back in the day before the snowstorm. When you see an answer and it looks right and you pick it, don't change your answer at the end of the test. Unless you have a really good reason of why the answer you picked is wrong and why a different answer is right, don't go back and second guess yourself. I always like to tell students, go with your gut. So if you think it was right, it's probably right. If you think it was wrong, it's probably wrong. Don't change those answers. And I always tell students too, don't lose your head. If you hit a question on the exam and your mind is completely blown and you have no idea how to answer that question, skip it and come back to it, because I guarantee there's at least 35 or 40 other questions on the exam that you do know the answer to. So come back to any ones that are blowing your mind. You've got plenty of time. Read things slowly and carefully. Don't second guess yourself. So Anthony's asking about the format of the exam. It's going to be very similar to your homework assignments. So there'll be some multiple choice. There'll be some that are that drop-down menu like you're used to doing, where the answer choices are embedded in the sentences. There's not really going to be any, I don't think there's any formats that you haven't seen before. So I'm not going to throw something new at you on the exam. But there's not any written questions, put it that way. Yeah, Kaylin says she's feeling a little bit nervous. That's understandable. It's your first exam with me. What I would say is use those practice assignments, do them as many times as you can, and do them without your notes as much as possible to kind of mimic what that test environment will be like. The more that you can pretend that you're taking the test, the more it's going to feel normal for you when you go to actually take it. And if you feel like you're losing your head, your mind's getting blown while you're taking the test, pause for a moment, close your eyes, take a deep breath, and then start again. So you've got plenty of time to go as slowly as you need to take those moments that you need to. Yeah, so Kaylin, if you're always getting about the same score, the goal with that assignment isn't necessarily to make you have to go get a 100, as much as the goal of it is to help you find what topics you still need to study. So if you're missing the same kinds of questions every time, that helps you know what to study. If you're missing different ones, that helps you know that maybe we need to slow down and make sure we're focusing on the right stuff. So you've got plenty of time on that assignment and plenty of time on the exam. Use it as a learning tool, not just as a grade, but try to see if you can use that to help you figure out what topics we don't need to study anymore, the ones that you're always getting right. Ignore those topics, we know those well enough. Let's focus on the stuff that we don't like to study as much because we don't know it as well, right? We all like to study the stuff we know. The stuff that we don't know is the stuff that we need to spend more time on. So figure out what you're missing and then focus on studying those things to get you ready for the exam. And the good news is, at the end of the semester, we take a final exam that includes stuff from each of the units during the semester. I actually replace your lowest exam score with your score on the final. So if exam number 1 doesn't go well for you, it's not the end of the world, because at the end of the semester, we can replace that with your final exam too. So don't freak out too much. Don't lose your head. That's going to be a way to forget what you studied. So give yourself some grace, keep studying, take a break for a little bit and come back to it with a fresh mind. And again, it's not the end of the world. It's one test and we will move on from it. Yeah, so I like that attitude, Anthony. We're going to keep practicing. I love it. And Anthony's asked about the lab exam. So the lab exam is still next week, but it's at the end of next week. And we'll talk more about that tomorrow. During tomorrow's class, I'm going to do lab stuff. So we're focusing right now on the lecture stuff because we've got that lecture exam upcoming. Only week 5 is due this Sunday for lab. So the lab homework from week 5, from the week that we missed in the snowstorm, is due Sunday. Everything else will be due next Friday, or next Sunday, excuse me, when the midterm exam is due for lab. I moved the lecture assignments that were due on Sunday of this week. I moved those to next Sunday. So lesson number 5 stuff is due next Sunday. This is actually a great question and a good time for me to mention that there is an updated assignment checklist. Please download that and print it, because unit number 2 and some of the lab stuff in the next few weeks is going to be a little bit different. Nothing is due earlier than it was going to be due before. So if you keep using the old assignment checklist, you're just going to get stuff done early and you'll be good. But if you want those new dates, that new assignment checklist is posted for you. So check that out to reference new dates for lecture and for lab. Yeah, Kaylin, we will do a whole histology review session for lab. So don't worry too much about histology right now. Focus on the lecture stuff and know that we will definitely have a histology review session for sure. Yeah. All right, well, I'm going to go ahead and turn off the recording. I will stick around for a bit for any questions that you have. Otherwise, good luck on the exam. Don't forget to breathe. I believe in you. Oh, here, let me draw you your penguin. Oh, my computer is dying, so maybe I won't draw your penguin. Ooh.